So we got a lot of things here to talk about and discuss. Let's go ahead and just start by getting the vocabulary words out of the way. We've got genotype, we've got phenotype, we've got dominant, we've got recessive. Okay, genotype, phenotype, dominant, recessive. All right, so the genotype represents the alleles at a genetic locus that result in the phenotype. The phenotype is going to be the physical trait observed as a result of the genotype. So, what that means is if the genotype up here is a big B and a little b, and that results in brown eye color, we would represent the genotype as the two alleles that contribute to the overall physical characteristic. In this case, that is brown eye color. So that's the difference between genotype and phenotype. Dominant and recessive come into play when you're comparing those two alleles. So the dominant allele is the allele that results in the observed trait. More specifically, if that allele encodes a distinct trait, that trait is what winds up showing through in the phenotype. So for example, here, if big B represents brown eyes and little b represents blue eyes, then the observed trait is based off of that big B, which would make, in this case, the big B the dominant allele. Now understand and remember that dominant and recessive are relative terms. If I all of a sudden pair up that big B with another allele, and the other allele winds up expressing its trait, or that trait of that allele is expressed, then that means that this is no longer dominant. Okay? While the recessive is the allele that is not expressed. Okay, so you don't see the trait of the recessive allele. Okay, so there are two other terms we need to discuss here, and this is the perfect way to discuss them. Those terms are homozygous and heterozygous. And remember, if I'm referring to an individual who is homozygous, I can call that individual a homozygote. If they're heterozygous, then I can refer to them as a heterozygote. Okay, so homozygous, heterozygous. Homozygous means the individual has two of the same allele at a genetic locus. That means big B, or little b. Those are both examples of homozygous individuals. A heterozygote, or heterozygous individual, has two different alleles at a locus. And that's like the example up above, where you get one dominant, one recessive allele. So, I might again not just ask you for general definitions of these, I might ask you a question that pertains to all of this, where I just use these, the understanding that you're going to understand what I'm saying. Okay? So just be comfortable with these vocabulary words. All right, so now let's go ahead and start looking at some Punnett squares and start determining the likelihood that we'll get distinct genotypes and phenotypes. And we're going to do a monohybrid cross, and then we'll talk about a couple of exceptions to the rules when it comes to dominant and recessive phenotypes and genotypes. Okay, so let's first do just a really basic one. Let's do a monohybrid cross where we have a father who has, let's say, brown eyes, and his genotype is big B, little b, and a mother who also has brown eyes, which will be big B, little b. Okay, so first and foremost, before we even go anywhere with the Punnett square, questions I could ask you pertaining to this. Which allele is dominant? Brown eyes or blue eyes?
Right, brown eyes.
Brown eyes, right? Because both the mother and father are heterozygotes, and they both have brown eyes, which means the brown allele, the big B, is dominant in this particular instance. Now, that kind of already answers the next question, but I could ask you, would these individuals be classified as homozygotes or heterozygotes? And the answer is hetero, because they've both got a combination of the dominant and recessive allele.
So if they both had big B, it would be homo.
And if they both had little b, right? So either one of those, if they're two of the same, because that's what homo means, is the same, then they would be homozygotes. Okay? All right, so let's go ahead and cross them. So we're crossing a heterozygote with a heterozygote. So we need to set up our Punnett square. So the way that we're going to do that, we're going to pull the big B here over to here. And we're going to pull the little b over to here. And then we're going to pull this big B here, and this little b here. And then we just write whatever is for each quadrant. I've got two big B's. I've got a big B and a little b. I've got a big B and a little b. And then I've got two little b's. Okay? So, questions I could ask you about this. First off, I could ask you for the relative percentages of genotypes. So for genotypes, let's talk about the percentages of each genotype that we could obtain from this particular cross. Okay? So what's the likelihood I would get big B, big B?
25.
It's 25%, and if I were to write that as a fraction,
1 over 4.
1 over 4, right? 1 out of the 4 squares gives me that. Okay? What about big B, little b? 50%. Okay, represented again as a fraction.
2 over 4.
Right, 2 out of 4, which I can reduce to 1 over 2. So 1/2 of these. And finally, little b, little b.
25%.
25%, which again is 1/4. Okay. So, homozygote for the dominant allele, heterozygote, homozygote for the recessive allele. All right? Now, phenotypes. Let's talk about the potential phenotypes that I will obtain. There are only two. Brown eyes and blue eyes. Okay, what's the percent chance I will get a child with brown eyes from this? 75. So understand, if we're talking about what's known as complete dominance, one allele is dominant to the other,
Right.
then the phenotype for the dominant allele will be a combination of the homozygote for dominant and the heterozygote. So here we see 25 and 50% gives us the 75% for the phenotype. Okay. And then the other, that one's 75%, then blue eyes has to be 25. So this is 3/4, This is 1/4.
On the test, will it be in fractions or percentages?
It can be either or.
Okay.
Yeah, it can be either or, fractions or percentages. I tend to tell students to leave it in fractions if you're doing the math,
Right.
because it'll make it easier to come out with the answer. And when I say that, what I mean is, remember, one of the questions I can ask is, what's the likelihood that this father and this mother will have a child with blue eyes and is a girl? Okay? So if I ask you and use the word and, you're supposed to do something versus if I use the word or. Multiply, right? So if I ask you something and there's an and in the statement, the way that you would calculate that is you take the percent chance or the fraction of getting a child with blue eyes. So for and, it would be the percent chance or fraction for getting a child with blue eyes is 1/4 times the percent chance or fraction that the child would be a girl, which is 50%, okay, which is 1/2, and that gives me 1/8. Okay. Most of the time with these, I'm going to leave it in the fraction form and not ask you to convert it over to percents, because that starts to get kind of crazy.
Can you explain that again? The last one?
This one? Okay. So because I'm saying and, both conditions have to be met for this to be true. Which means not only do I have to get a child with blue eyes, but that child also has to be a female. Okay? So the chance of getting a child with blue eyes is 25%, or 1/4. But I also have to get a child who is female. And remember, there's a 50% chance that the child will be male versus female, because the mother gives an X chromosome. The father gives either an X or a Y chromosome. And there's an equal likelihood the father can give either one, which is why it's 50%. So this is the chance right here of getting blue eyes. This is the chance of it being a girl. Okay? And I have to multiply them because both of these conditions have to be met for me to fulfill what I asked here. Now, if I ask you for the same thing, but instead of using and, I use the word or, that means either one is acceptable. If the child has blue eyes, it's a win. That answers the question. If the child is a girl, that answers the question. Because I'm not asking for a girl with blue eyes. I'm asking for a child that either has blue eyes or that's a girl. And in that case, whenever you're doing or, what do we need to do? You add them together. So 1/4 plus 1/2. If I bring this up to 2/4, that gives me ultimately 3/4, or 75%. So notice when I use or, I'm far more likely to get the outcome I'm expecting, because I'm using two criteria instead of one. Okay? I mean, if it makes it easier to think about, think of an or statement like this. What is the likelihood that the child will be a girl or a boy? It's 100%, because each of those outcomes is 50%. And as long as one or the other is obtained, it makes the statement true. Okay? All right, so that's the rules of addition and multiplication when we're talking about probabilities. Nobody likes them, apparently. All right, so let's talk about exceptions to the dominance rule real quick. And then we're going to jump into talking about linked and unlinked genes, and then we'll do an example of a pedigree together so that y'all are all comfortable with those. Okay, so we learned about two different things in regards to dominance that were exceptions to complete dominance. We had what's known as incomplete dominance. And then we had what's known as codominance. So let's go ahead and talk about these two. So with incomplete dominance, neither allele is dominant. So let's again look at a particular scenario of this. Let's say we have a father who has brown eyes, and we have a mother who has blue eyes. So we do a monohybrid cross with those two. So here's my Punnett square. I'll take the father. It's big B, crossed with little b. In this particular instance, they can be in either order. It doesn't matter. I can do big B, big B, little b, big B, doesn't matter. And let's say that when I do this, so first off, 100% of the offspring in terms of the genotype are big B, little b. For my genotype, I have 100% that look like that. That's not really where you determine if it's incomplete dominance. Where you determine if it's incomplete dominance will be with the phenotypes. So let's say that when this happens, 100% of the children have hazel eye color, rather than brown or blue. Neither of the two alleles is dominant to the other. In fact, the phenotype appears to be a combination of those two. Hazel eyes are brownish colored eyes with other colors infused with them, greens or blues. So this is where I'm seeing the incomplete dominance. I'm not getting a color that is one or the other, I'm getting a color that's a combination of the two. So to determine if something's incomplete dominance, you're going to have to look at the heterozygote. The homozygotes won't tell you one way or another. You have to look at the heterozygote to determine if it is incomplete dominant. Okay? Now let's talk about codominance. This is different from incomplete. Notice with incomplete, there was no recessive allele. With codominance, there's going to be a recessive allele, but there are going to be more than one dominant allele. And the example here, so we have multiple dominant alleles, and we have a recessive allele. That's what makes this codominant rather than incomplete dominant. And the example we looked at in lecture and you looked at in lab was blood typing. So blood type is an excellent example of this. So let me go ahead and show you that. I'm going to erase incomplete dominance so we can talk more about codominance. So with codominance, rather than picking a single letter, because you'll know in blood typing you have type A and type B along with type O. We don't want to pick a single letter to represent the different blood types like we did with eye color, where it was big B and little b. We're still going to pick a letter, but we're also going to denote that letter with a superscript if it's one of the dominant alleles. And for blood typing, traditionally, the dominant allele for this is going to be a capital I, but it's going to have one of two superscripts. It's going to either have a capital A, or it can also be a capital I with a superscript of a capital B. This denotes A-type blood. This denotes B-type blood. And the recessive, since we're dealing with I, is going to be a lowercase I. And this represents O-type blood. So what this means is as follows. If I have two big I's with the superscripts A, I have A-type blood. If I have an uppercase I with with a superscript A and a lowercase I, this is still recessive. This is still dominant, so I have A-type blood. If I have the same scenario with the big I's and the B's, it's going to be B-type blood. And again, if I have this, it's going to be B-type blood. And if I have two lowercase i's, they're both recessive, I'm going to have O-type blood. None of these demonstrate fully the codominance. What finally kind of rounds this out in regards to the codominance is this. When I have the two dominant alleles together, neither is dominant against each other, okay? The other. So I would then have AB type blood. So the allele for A is dominant to the allele for O. The allele for B is dominant to the allele for O. But neither A nor B is dominant towards one another. They're both expressed on the surface of those cells. So this is why blood typing is codominant rather than incomplete dominant. Okay. Any questions regarding that?
Well, on the test, would you ask, like, is O like the donor or?
Okay, so in terms of blood typing transfusions.
Yeah.
I can potentially ask questions about that, so let's just discuss it real quick.
Okay.
So the rule of blood typing is if your cells don't have it on their surface, they produce antibodies against it. So A-type blood produces antibodies against B-type blood. So they make anti-B antibodies. What this means is if a person with B-type blood tries to give to a person with A-type blood, that blood will be rejected. With B-type blood, the individual makes antibodies against A. Remember, these A's and B's, those represent carbohydrates on the surface of these cells. So what's going to happen if an individual with A-type blood tries to give to a person with B-type? It's going to be rejected again, okay? The body will not accept it, okay? Next, O-type blood. Do cells with O-type blood have either A or B on their surface? No. So what are they going to do? They're going to make anti-A and what else? Anti-B. And that means if a person of either of these two blood types tries to donate to a person with O-type blood, what's going to happen?
You're rejected.
They're going to reject it, right? Okay. But what does this mean in terms of O-type blood giving to A or B?
Yeah.
Right. O can give to either A or B, which is why O negative is commonly viewed as the universal donor. Okay? So O-type blood can donate to both A and B. All right, and finally, AB blood. Who can it accept from?
Both.
Both. It has no antibodies. So it can take from A-type blood, B-type blood, and O-type blood, which is why this is commonly viewed as the universal acceptor, because all three other blood types can donate to AB. But can AB donate to any of the other blood types? No, it cannot. So AB can't donate to anybody else, but it can accept from everybody else. It can obviously donate to itself, and that's a general rule with all of these. A-type blood can donate to A-type blood. B-type can donate to B-type. Okay? All right, so that's blood typing. Any other questions regarding that?
Will you have the pictures?
Will I have the pictures like the actual blood cells? I might. Yeah, I might use those blood cells to talk about the carbohydrates on the surface. If I don't give you the picture, I'm going to give you an adequate description of the particular blood cell in question.
Okay.
Okay?
Did you mention what the question is going to look like?
What's that?
Did you tell us how you're going to ask the question on that?
On this one, I'm either going to show you a picture of the red blood cell and say, what blood types could this donate to, or what are potential genotypes for this, or what blood types could it accept from. Or I could give you a description of the cell and say, a particular red blood cell is, has no carbohydrates on its surface in regards to A and B, who can it donate to, who can it accept from? Or I could just give you the genotype and ask you, what kind of red blood cell is this? Is it A type, B type, AB type, or O type? I can also ask you, or tell you, any of these, and then just ask you a general question about blood typing, and say, is this an example of codominance, incomplete dominance, or complete dominance? And if it's dealing with red blood cells, we're talking about codominance. Okay? All right. Why don't we finalize this review session with two last things. I want to talk about linked and unlinked briefly, because there's only one thing you really have to keep in mind about that. And then after that, we'll do a pedigree together, so that we're all kind of on the same page with pedigree. So, let's talk about linked and unlinked. So remember, this was determined with that dihybrid cross that Mendel performed. The monohybrid was looking at one trait, while the dihybrid was looking at two. So the example they gave in the book, remember, was round yellow peas crossed with wrinkled green peas. Okay, so first thing to note was that they told you, so we're looking at two different traits. We're looking at shape and we're looking at color. And in the book they mention the fact that the yellow and the round are the dominant. So this is dominant, this is dominant, this is recessive, and this is recessive. Okay? So then when we cross these, and we get our F1 population, the entire F1 population was round and yellow, and that's also how we know that those are the dominant ones. And then what happens is we allow these to self-pollinate. So they self-fertilize. And the end result in the new population is we get 4 distinct phenotypes. Okay? Those 4 distinct phenotypes are as follows. We get yellow and round, just draw here, round yellow peas. We get wrinkled yellow peas. We get round green peas. And finally, we get wrinkled green peas. Okay. Now, I'm just going to go ahead. We're going to mark real quick whether each of these traits is dominant or recessive. So remember, round was dominant, yellow was dominant, wrinkled was recessive, yellow was dominant, round was dominant, green was recessive, and then wrinkled was recessive, and green was recessive. So our 4 phenotypes are one of the original parental strains, the other original parental strain, and then 2 combinations of the original parental strains. Now remember, this represents the dominant parental strain. This represents the recessive parental strain. And really the big thing that I wanted you to remember from all of this was the ratio in which each of these occurs. Okay? Does anybody remember what that ratio was? 9:3:1. So if we were to have an ideal situation where we made 16 of the potential offspring from the percent chances of each, we would find that 9 of those 16 would have a chance of being round yellow, 3 would be wrinkled yellow, 3 would be round green, and 1 would be wrinkled green. So this gives you a general potential for each of these different genotypes / phenotypes. Okay? And actually these are all phenotypes. Okay, so this is out of a total of 16, and if you get this number, can you remember, are they linked or unlinked?
Unlinked.
They're unlinked. If you get this ratio, you're dealing with unlinked genes, which means what that tells you is the allele or locus for the round or the shape of the P is far away from the allele that encodes the color of the P. If they're really close together, you're going to see round and yellow associate with one another constantly. You're going to see wrinkled and green associate with each other constantly. And you're going to see these two combinations decrease in their total amounts. Okay? So, if I give you a question, it's probably going to come out close to a nice 9:3 to 3:1 ratio. And if it doesn't, then you should immediately be thinking that they are not unlinked, instead they are linked. Okay? So this tells you that they're unlinked. If you don't get that ratio, they're linked. Questions, concerns about this particular aspect? All right, let's close it out with a pedigree. And then I'll take final questions if y'all have them, and if y'all don't, then we'll be done.
Can we ask you a question about the quiz?
Sure, if you'd like to.
I'll wait then.
Okay, so I'm going to draw the pedigree first, and then y'all will help me kind of figure out what's going on with this. So, let's go ahead.
Oh, crap. Forgot about this.
That's why we're going upgrade.
It's like a family tree.
Pretty much,
Yeah.
yeah. Okay, so here's my pedigree. And let's start by doing this. Okay, so first off, let me give you your key. So your key is as follows. For males, you're going to see this. Females, you'll see this. And then if the individual has the disease, or whatever the condition is, they will be shaded. And the final thing I will tell you is that this disease, whatever it is recessive. Okay, so, knowing that, I want to know what the genotypes or potential genotypes are every person on this pedigree. So, let's start with the easy ones first. And let's start first by just deciding a general letter that we're going to use to denote the disease. Why don't we just do, since it starts with a D, we'll just do Ds. Okay? So we've got 3 potential combinations. Okay? Which of these 3 combinations represents a person with the disease? If it is recessive. Is it going to be 2 big Ds, a big D and a little d, or 2 little Ds? 2 little Ds, that's our recessive. Okay? So, there are 3 people to start off with that you all can tell me in regards to their general genotypes. Okay? What are those 3 people? Any one of the shaded ones. I know each of these people who are shaded have to have 2 recessive alleles. Okay? Now, for these 2 people, based off of the child they have, I know for a fact one of these came from each parent. So, what's one of the alleles going to be for each of these parents? A big D or a little d? A little d. I know this parent's going to be a little d, and I know this one's going to be a little d. Okay? Next part of this. are either of the parents shaded? No. So what does the other allele have to be? Big D. Okay, everybody see that?
Yeah.
If it was not recessive, then it might be another issue altogether. But because this child has two little d's, one of those has to come from each parent. And because neither of the parents are shaded, I know that these two parents have to be heterozygotes. That make sense? Okay. All right. Now, let's do the same thing for this child. What do these two parents have to be? Okay. They have to have a copy of the recessive, and what do they have to have as their other allele? Big D, the dominant, right? Because they don't show the shading denoting that they have the disease. Okay. So if you look, we've already filled in almost every single person on this entire pedigree. The last two are going to be the ones that we're uncertain about. Okay? Is there a way for me to say for a fact that this person is either two big D's or a big D and a little d? Can I safely say?
No.
I can't. I don't know. I know for a fact it's not little d because it's not shaded, but I don't know if this person is a homozygote for the dominant trait or a heterozygote. And on a question like this, a completely acceptable answer would be
No.
or that. Okay? Or, as you were saying, Hope, if I ask you, can I be certain that it is this or this, the answer is no. Now, if this person, and I'll show you in a second, there's a scenario where I could see if this person was this one or that one. Okay? And I'll show you in just a second. Okay, next. This person. Can I safely say this mother was a homozygote for the dominant trait or a heterozygote? No. Again, I don't know. Because they only had one child. So again, I can't be certain. I know that the mother doesn't have this, but either one of these scenarios could have given this child a big D. Does that make sense?
Yeah.
Okay. Now, two scenarios that, because I might give you a follow-up question, that could help you determine the genotype of this person or this person. If these two parents had more than one child, let's say they had two other children, okay? And one of these children had the disease, what does the mother's genotype now have to be?
Yeah.
Big D, little d. She has to be a heterozygote, right? Everybody sees why?
Right.
This person here is two little d's. The only way this person could get two of those recessive traits is if the father gave one and the mother gave the other. Okay? For this one, to determine that, I'm going to erase my key real quick. The way I would know is if this person were to have children with the person who has the disorder, or the disease, and the child they have, if the child also has the disease, what does that tell me about that mother?
Big